Thank you. The game of COVID simulations, team one. Team members are John Paul Robinson, Devin Kale, I hope I'm not pronouncing those names wrong, and Kamal
Cole.
Okay, Cole.
Yeah, so we just looked at the problem of epidemic modeling with a tool called a cellular automaton, which is popularized from a mathematical game called Conway's Game of Life. If we can go to the next slide. The paper that we largely based this work on, or the motivation for this work on, was a paper by Gilpin that from last fall that basically explores the cellular automaton and rule learning from a neural network perspective. So it tries to use a convolutional neural network, which is really good at two-dimensional structures. It's used heavily in image recognition, and basically treats the game boards for Conway's Game of Life as a 2D image, and then learns the image transitions from one generation to the next. If you'll see, there's a game board that's on the left-hand side of these images, and the game board has various transition rules that we'll go over in just a second, and each generation shows what happens on the next generation. So if we feed in a sequence of these images, we can potentially learn what the transition rules are without specifying them in code. We'll go to the next slide.
Okay, so I'm going to talk to you about Conway's Game of Life and the rules. So the Game of Life is a cellular automaton, and it's devised by British mathematician John Conway. It's a well-known application in computer science because of the various rules that govern each state. The Game of Life is played on a board, as you can see here, and where each state can either be alive, the black cells, or dead, the white cells. And so the rules that govern the Game of Life are as follows. A living cell will die if they have fewer than 2 neighbors. This is called underpopulation. A living cell dies if they have more than 3 neighbors, and this is called overpopulation. Dead cells that have 3 neighbors become alive, which is called reproduction, and then otherwise there is no change to the system.
And one of the things that makes a cellular automaton so appealing is that through a very basic set of rules that govern just individual cells, you can actually model, or they take on the operation of complex systems. They're used in a lot of different areas of research. And so what we wanted to do was to basically take the COVID-19 infection data, positive results for infection, and take that as our input data set to build essentially Game of Life game boards for that. And at the county level, we were basically looking at just the county data, as you kind of see in this map here, build a game board for it, and then see if the sequence of generations that we would create, if on the next slide we can see that. Well, I guess on the next slide we actually have the data set, but we're just focusing on the county level data in the US, and which is kind of standard from all the data that's been published from Johns Hopkins. And in this particular case, we were working on the Kaggle data set that's further curated a little bit and summarized. But on the next slide, we can see the game board that we developed. We essentially take our infection data and we scale our county points on a geomap of the United States, and as time increases, we took each day of reporting as one generation of the map, and you can see that as the generations move forward, the squares get larger, and so you can clearly see where the epidemics are here in the United States, or the heavy epidemics. You can see New York, Seattle, a little bit in California on the edge, and then emerging in Louisiana and South Florida. Chicago and those kind of areas. So in this first rendition, we just basically said, okay, let's create game boards implicitly by just allowing the rate or the count of infections to scale our squares, and see if we can learn from that. Move on to the next.
Yeah. All right, so the steps that we used to process the training data, we read in all those images that you saw on the previous slide, converted them all to grayscale, and used this library called OpenCV in Python. We reshaped the images and then cast them as a NumPy array and divided them all by 255. So now we have all of these with, that are matrices of 0 and 1. So using this, we converted them to a tensor for our convolutional neural network. We then split the data into training and testing. And that Gilpin code that you saw at the beginning of the slide set, we used a function within that called make game of life, and it returns a simplified TensorFlow implementation on Conway's game of life. And so here you can see some results that we had from training. We used 50 epochs here, and we have this nice graph that shows the loss by epoch. And here are the model details on the left-hand side. On this slide, we wanted to show the activation of each of the hidden layers. The hidden layers of the CNN typically consist of a series of convolutional layers, and below you can see the convolutional filters. Okay.
And if you could go back to that previous slide for just a second, Devin. One of the things that we kind of want to note is in Gilpin's model, the layers are meant to essentially learn the activation rules for the neighboring cells. And so when you think about the pictures that we have where our squares are getting bigger, kind of the rule is very simple here. How fast does the square get bigger? And so that's kind of what we're seeing reflected in our current implementation of the CNN with the images that we have available to us at the moment. And now we can go on to the next one.
Sure.
And so the next round that we want to follow is actually improving the images that we generate so that we can learn hopefully much more complex and subtle aspects of, I guess, our game of COVID and how COVID actually spreads. There's another paper that we were looking at by White, and it essentially looks at how to model epidemics using cellular automaton. And one of the models that's used for epidemics is the SIR model, so the percentage of susceptible and infected and recovered members of the population. And so by using that model, we essentially can have a 3-value or 3-tuple value for each cell in our network, representing these 3 different aspects of the infection. And then that essentially allows us to start modeling a much more subtle growth of the infection across the network. It also requires that we have a maybe more mature game board. Our initial game board was simply the geometric or the geographic center of the county as reported by standard data sets. Actually, it's reported by the Kaggle data set included there. And so we just kind of started plotting our points there. But as we create a formal game of life game board, one of the things that we need to do is actually have all our counties adjoining each other in just a regular grid. And so we'll be using census data for adjoining counties in order to build that grid so that we can then include that. And the SIR model for the project that the paper studied several different configurations of mobility within the population. And so it has a lot of subtle parameters that can be tuned and learned. And we hope to be able to actually, instead of having to set those parameters, learn those parameters through our CNN. And let me see if there's any, in order to model this at the moment, we're assuming a recovery time of 2 weeks. So we don't have any really solid recovery data. So just to get that calculation in for the initial round, we had basically subtracted out the infected from 2 weeks ago from the infected today to give us a sense of who's infected, actually infected today. Because the SRI model nicely maps to essentially three distinct values, we can treat it as like an RGB image or enhanced grayscale.
Okay, so the summary of the hackathon for us was, cellular automata can be used to model a variety of natural systems, including epidemiological behaviors. Cellular automata are used to model COVID-19 data, and time series images are generated for infection by county. And so our question is, what insights could we gain from COVID-19 from being modeled by cellular automata? And lastly, we wanted to dedicate this to John Conway, who passed away on April 11th, 2020 due to complications of COVID-19. Thank you.
There's a question, how applicable is neighboring square model to a disease spread that is thought to be highly impacted by long distance travel, such as flights?
Well, so if we might go back to the white paper, the parameters that the white paper includes are actually include connectivity and mobility. And so they have parameters that you can specify, not in the equation that I'm showing here on this slide, but there's an additional term that can be included in the transition rules that model if the cells are connected by car, by train, or by plane. And so there's actually a decent way of representing that information in the models presented in white. And so the question, I guess, we have is that insight derivable from a visualized representation from a CNN network? If we kind of basically create these density models of our cellular network, can the CNN accurately predict that? In which case, does the CNN learn what those parameters are? In the paper of white, he just basically, the team there selected 3 different configurations of a roughly 1/3, since there's 3 modalities for travel and 3 modalities for movement that they used in their model. They just basically ran several different models with, saying 1, 2, or 3, all 3 are available. So it'll be interesting to see how that's reflected in real data of this epidemic.
There's another question. We're at 11.5 minutes now. So about county connections, were they included? Or?
I'm sorry, county connections or counting connections?
County, like counties?
Yeah, so in our present data set that we trained on with the squares growing, the connectivity is only implicit by the virtue of the fact that we used bitmap images, and so they naturally create a grid, and then that naturally maps to a 2-dimensional array. Unfortunately,
we have to stop because
okay.
time's up.